Welcome to our 10th video on the topic of mitosis. In our last video, we discussed the stage called cytokinesis. We went over some of the key points, including the key stages of cytokinesis and how cells separate their cytoplasm and split into two daughter cells. If you missed that video, please watch it before watching this one. In that video, I promised that we would discuss Anaphase spindle reorganization, division plane specification, actin-myosin ring assembly, contraction and abscission. In this video, we will focus on the first step, that is the Anaphase spindle reorganization, and we'll discuss that in more detail. In addition to that, in our previous video, we also discussed how animal cell cytokinesis is completed. Important points to remember are involving the stabilization of microtubules and reorganization of the spindle, mitotic spindle to form the central spindle. Now the question is, what exactly is the central spindle? The central spindle is composed of microtubules that is formed as the chromosomes are separated in opposite directions. Microtubule associated proteins, MAPs, and motor proteins organize the microtubules from each half of the cell, causing them to overlap and form bundles. Now let's move to the molecular mechanism of this particular process. As anaphase progresses, the activity of CDK1 declines. The phosphorylation occurs on several components of the central spindle. As a consequence, a subunit of the chromosomal passenger complex, CPC, moves from the centromere to the central spindle. Further, CPC governs the activity of other central spindle components such as PRC1 and MKLP1. PRC1 is a protein that helps bundle microtubules required for cytokinesis. It can form a homodimer that selectively binds to the interface between antiparallel microtubules. So if we want to summarize all these important points, the decline of CDK1 activity initiates the self-assembly of the central spindle by regulating multiple central spindle components directly as well as indirectly. It is important to note that the central spindle is responsible for controlling cleavage furrow, positioning, delivering membrane vesicles to the cleavage furrow, and forming the mid-body structure required for the final step of cell division. So these were some of the key points regarding the Anaphase spindle reorganization. There are so many things that are still unknown to this particular process, so researchers, they are diving deep into the biophysical understanding of this particular process. I hope you liked the video and understood the molecular mechanism of key processes, that is, in this case, Anaphase spindle reorganization. Okay, finally, one request from my side. If you liked the video and enjoyed all those videos and content, please do not forget to subscribe to the channel. Thank you. Let's move on to the next video.